assembly of a Y chromosome from a wild vertebrate, namely this male swordtail fish right here. So Xiphophorus is a long-standing evolutionary model. It has well-characterized genetics and phenotypes for key Y-linked traits like pigmentation and body size. And it's also been used as a model for human melanoma, so it's both inducible with UV and spontaneous, as in certain hybrids. So here's Xiphophorus nigrensis. This is a sort of well-known study where you can see you have a normal-sized male with a nice sword, and then you have sort of a dwarf sneaker male. And the difference between these two phenotypes is the number of non-functional MC4R receptor genes that are present on the Y chromosome. All right, so it's Xiphophorus platyfish and swordtails are an emerging genomic model. There are 3 currently published nuclear genomes, all female, and there are pseudogenomes available for 3 additional species, and transcriptome data for 24 of the 26 species. So we still, unfortunately, know little about the genomics of the male sex chromosomes, despite their importance in sexual selection and sexual conflict. So in our lab, we work not so much on platyfish as on these 2 swordtail species, the Highland swordtail, Xiphophorus malice, and the lowland or sheepshead swordtail, Xiphophorus bertrandi. You can see here a nice selection of male hybrids. You can see there's some good variation in pigmentation and sword presence or absence and body size. So lab crosses are fertile, but you really don't find early generation hybrids in the wild, and that suggests that there's strong premating isolation. So there's been a study that found lots of pairwise incompatibilities among loci in the 2 species. They're over here, you can see they're just spread across the genome. And given its role in resolving sexual conflict, the Y should be important to isolation, be its effects on targets for mate choice and into the evolution of genetic compatibilities. You can see, sort of depending on where you put the cutoff, there's 150 to 300 incompatibility pairs identified so far. So Xiphophorus sex chromosomes. So in general, we're lucky in this genus that the genomes appear to be pretty much collinear. The X and the Y, though, are homomorphic. The non-recombining region of the Y is not very big. It's estimated at a megabase. And there exists these back clones that span this sex termination region in platyfish. Unfortunately, the sequence is not available, but it was described at least. And there are these 11 genes that are found both on the X and on the Y in this region. So taken all together, homomorphic, not a very big non-recombining region, and genes present on both X and Y, these all suggest young for differentiation. So we sequenced a male and female from malice and a male bertrandi. We used Discover de Novo. It's a pretty good program. If you're curious about that, talk to me afterwards. We scaffolded it with some jumping libraries, and we basically just did this scaffolding, gap filling, breaking of disassemblies process 3 or 4 times until the N50 started to plateau. So the male genome has an N50 of 800 kb. Female genome's a little better. And they're both close to the estimated genome size for this entire genus, which is about 750 megabases. We estimated the completeness of the genome using SEGMA. It's this set of core eukaryotic genes. So you can see ours is sort of the best we looked at. It's marginally better than Medaka. And we have almost 99% of the conserved orthologous genes present in at least a partial copy. So yeah, we don't really know what's on the Y. There were those 11 genes in platyfish that are supposed to be on the X and Y. I looked for those first. I didn't find that helpful. I looked at coverage. That didn't work so well. So I ended up using this chromosome quotient method that Brantley Hall devised. And basically, you take a male genome assembly, and you map back the raw reads to it, and you take female raw reads and you map that to the male genome. And you compute this chromosome quotient, which is the number of female matches to the number of male matches. These are actually perfect matches. So it's sort of like coverage, but not exactly. It allows you to distinguish between things that are basically 97% identical. And I've gotten the CQ values for all the de novo contigs, and I log transformed them, so you can see the nice left-hand tail. That's the tail we're interested in. And anything with a CQ of less than 0.3 is iteratively Y-linked. You can see this sort of shaded region would contain about 12 to 1500 putative Y-linked contigs. That's too many to do anything with. So I ended up running the same script on the scaffolded assembly, and I came up with 20 some odd scaffolds. Unfortunately, more than half of those were more than 50% gaps. So that's not much to work with. So I ended up with 8 scaffolds that were larger than 25 kb, so arbitrary cutoff, and less than half gaps. And conveniently, they add up to almost a megabase, which is right the estimated size of the non-recombining region in platyfish. So the repeat content of these is very high, and the sequence identity with the female platyfish genome is pretty low, except within some protein-coding genes. That's sort of what you might expect to find on a Y chromosome. So let's just look at one scaffold briefly. This is probably the most interesting one. So this is just a blasting my scaffold against the female platyfish assembly. See, there's some really large regions with just nothing that's even remotely similar to the platyfish genome. This is a gap in my scaffold, so that doesn't really count, but there's no gaps at that end. It just doesn't match very well. And you've got some stacked blastids, or common repeats. But this is actually sort of the interesting end of the scaffold, because this contains, you can see it's well covered, but not all in one hit, right? It has sort of a patchy coverage, and these are all genes that have been moved from what is in platyfish an autosome, linkage group 5. And there's actually some pretty interesting genes in there that we'll get to talk about. I will say that we have a collaborator who's also working on the platyfish Y chromosome at the same time, and he has this region, but in a lot more pieces than we do, and he has scaffolds that correspond to the sort of right-hand side of this 107 kb scaffold. He doesn't have what's on the left-hand side, which we're going to talk about. So I took some of these larger repeats on the 8 scaffolds, and I ran them through the conserved domain database. Certainly, a lot of them are transposable elements, but not all of them. Some of them are clearly derived from nuclear genes. So, for example, I find these 3 domains in a row, the noctin, fish-specific noctin, and leucine-rich repeat, find those 3 domains in a row on 3 of the 8 scaffolds. So this sort of suggests that these scaffolds contain the same junk that's being sort of shuffled around, which further suggests that they're all the same thing, they're all Y, and so on for the rest of these domains. All right, so I told you that at the left-hand end of that blast diagram, there were genes from an autosome in platyfish, and these are 2 of them, and they seem like they could be candidate sex termination genes. So GAS is expressed in testes in mouse, and it's absent from infertile human males who lack the ability to produce gametes. All right, that's sort of far down the sex termination chain, but it's still male function, right? And this is steroid dehydrogenase, 5-alpha reductase, converts testosterone to dihydrotestosterone, and deletion causes pseudohermaphroditism in human males. So, even if these aren't the master sex termination gene, they're interesting as they relate to male function and finding their way to the Y. So, some preliminary functional evolutionary insights. So it looks like the platyfish and these Apocryphus Malinche Y chromosomes are homologous in the non-recombining region, but still divergent. And it looks like Malinche has been acquiring sequences that are found on an autosome in platyfish. So the Y-linked scaffolds look like they're evolving rapidly under relaxed selection. Lots of reshuffling going on. And we expect to find male function and sexual conflict genes. We do find male function genes. I think it's a little early for the sexual conflict genes. And we would expect the Y chromosome to play a major role in genetic compatibility. And that is something we're going to investigate further in a second. So, yeah, I showed you the sort of diagram of all the 150 or so hybrid incompatibility loci that we found in these natural populations. And we're going to reanalyze that data set now that we have a Y marker. And that should give us a lot clearer picture. We're also going to look for signatures of integration of Y-linked genomic regions in natural hybrid zones. I'll refer you to a couple talks that will address these things in more detail at the end. And we're going to try to improve the scaffolding of the Y and of the MC4R alleles with some nanopore data. I don't have much of a story to tell you about the MC4R alleles that make the body type difference I showed you at the beginning, simply because there's just a ton of full and partial copies, and they just didn't scaffold well. They're too similar to one another. But with these super long reads, I think we should be able to resolve both the non-recombining region and the MC4R alleles. Really, the non-recombining region shouldn't be more than about three long nanopore reads across, so it's definitely doable. All right, so I didn't get to talk much about what the lab actually does, which is hybridization mate choice. So if you want to know more about mate choice, please go see Pablo or Dan or Gil's talks. And hybridization, see Gaston or Molly's. And reproductive isolation, go see Chris Collins' talk. All right, thank you for listening.